Hello students, today we are going to start with a new chapter, that is anatomy of flowering plants. So anatomy is the branch of science that deals with the internal structure of organisms, that is called anatomy. So in this anatomy of flowering plants, we are going to study about tissues, structure of these tissues and the orientation, organization of these tissues inside the plant organs. And here we are going to deal with the anatomy of plant organs, that is dicot plants and monocot plants. Okay, let's start with it. So here, tissue. So in this chapter we are going to study about tissues, types of tissue systems, anatomy of dicots and monocots. Anatomy of dicots and monocots is nothing but anatomy of dicot root, monocot root, dicot stem, monocot stem, dicot leaf and monocot leaf. So anatomy of dicots and monocots. And secondary growth. So we are going to study about this chapter and these subtopics like tissues, types of tissue systems, anatomy of dicots and monocots and secondary growth. Okay, so tissue. The term tissue is given by, let's start with tissues. The term tissue is given by the scientist group. So tissue is nothing but it is a group of cells of common origin and common function. Common origin means they are originated from common point, common cell. That is common origin. And all these cells are involved in performing the same function. So common function. So a group of cells performing cells with common origin and function. This is called tissue. So these tissues are basically divided into two types on the basis of their capacity to divide. On the basis of their ability to divide, ability of cell division, these tissues are ability to divide. On the basis of their capacity of cell division, these tissues are divided into two types. They are meristematic tissues and permanent tissues. Meristematic tissues and permanent tissues. So meristematic tissues are the tissues which are able to undergo cell division. They are called meristematic tissues. Then permanent tissues. Permanent tissues are the tissues which cannot undergo cell division. They are permanent tissues. Okay, let's start with meristematic tissues now. Right, meristematic tissues. So meristematic tissue is the tissue which is formed by a group of meristematic cells. Meristematic cells are the cells which can undergo cell division. So that is called meristematic tissue. Now let's study the characteristic feature of meristematic tissues first. Characteristic features of meristematic tissues. Characteristic features of meristematic tissues. Right. So these meristematic tissues have the ability. They have the ability to divide. They have the ability to undergo cell division. And these meristematic tissues are made up of thin-walled cells. So the meristematic cells are smaller in size, small-sized, and these cells are thin-walled, and they are packed tightly. tightly packed cells. That means there are no intercellular spaces inside the meristematic tissues. Tightly packed without any intercellular spaces. Packed cells which are without any intercellular spaces. Intercellular spaces are absent. Okay. And these meristematic cells, smaller size involved, but they consist of dense cytoplasm. That means thick cytoplasm, more amount of cytoplasm. So they consist of dense cytoplasm and a large nucleus, large sized, prominent, large or prominent or conspicuous, everything is same. So they consist of dense cytoplasm and large or prominent or conspicuous nucleus. Conspicuous means the nucleus which can be seen very clearly. That is called as conspicuous nucleus, large nucleus, prominent nucleus. Okay. So these cells continuously undergo cell division, meristematic cells. To undergo cell division, they require more amount of energy, that is in the form of ATP. So where this energy is released, in which cell organelle this energy is synthesized? Mitochondria. And hence, as these cells require more amount of energy, the number of mitochondria present in these meristematic cells will be more. So meristematic cells consist of more number of mitochondria in them. More number of mitochondria. Mitochondria are present. Okay, next coming to storage bodies, or next coming to food, whether they are involved in synthesis of food or not. They are not involved in the synthesis of food. They do not synthesize food. So where from they get food? Where from they get means, so these meristematic cells, I am writing it as MC, meristematic cells do not contain chloroplasts. So chloroplasts are absent in these meristematic cells. So chloroplasts are a type of plastids. Here plastids. So plastids are of three different types. What are they? Chromoplasts, leucoplasts and these chloroplasts. So plastids are absent. But proplastids are present. So proplastids are precursors of plastids. What are these proplastids? They are precursors of plastids. Proplastids are involved in the formation of plastids. In case of meristematic cells, proplastids are present, but in case of permanent cells, plastids are present. These plastids are forming from proplastids. Hence, these are the precursors of plastids. Okay, so these meristematic cells does not contain chloroplasts, plastids are absent, and hence they cannot synthesize food. So food synthesis is absent. Food synthesis is absent. So where from they are going to get the food? So they absorb food from surrounding cells. Absorb food from their surrounding cells. From surrounding cells. And these cells are with greater absorption ability. Why? Because these cells are smaller in size. If the cell is smaller in size, the surface area of the cell will be more. If the surface area of the cell is more, it can absorb the nutrients at faster rate and with higher efficiency. So they can absorb nutrients or food from its surrounding cell as their surface area is high. Okay. So they absorb from surrounding cells due to their high surface area. Surface area is high. Okay. Next coming to some other characteristic features of these meristematic cells. And in these meristematic cells, vacuoles are absent. Vacuoles are absent. And even ergastic substances. What are ergastic substances? Secretory substances. Gums, resins, tannins, these are called ergastic substances. These ergastic substances like gums, resins and tannins are also absent. Ergastic substances are absent. And even storage bodies. Food is not synthesized and it is not stored. Whenever these meristematic tissues are in need of food, they absorb this food from the surrounding cells as they consist of high surface area due to their smaller size. So here storage of food is also absent, or we can say storage bodies are also absent. So these are the characteristic features of meristematic tissues or meristematic cells. These meristematic tissues are divided into different types based on their origin and based on their location.